Hello, and welcome to another episode of Mall Bio Review. I'm your host, Ryder, and today we'll be looking at two papers from the recent biotech literature. The first one's a study published in Nature Biotech this year in May. It's entitled DNA-Guided Genome Editing Using the Natronobacterium Argonaute. It's by a young man by the name of Gao Feng, and he's in the laboratory of Dr. Han Chunyu at the University of Zhejiang in China. In this paper, they proposed the use of a new Argonaute protein, a bacterial Argonaute protein, in order to affect genome editing, and they mainly tested on mammalian cells. Figure 1. In Figure 1, they test whether the guide for Argonaute is RNA or DNA, and they find that it's single-stranded DNA, and furthermore that it requires 5' phosphorylation to be effective as guide. Let's have a look at the gels. Here, using a nuclease digestion assay, they show that it is sensitive to DNase but not to RNase, thus showing that it's a DNA and not an RNA that's the guide. The second part, they show that their Argonaute protein is effective when used with one guide. It creates nicks in the DNA, but when they use both guides at the same time, they're able to linearize their plasmid completely, thus showing cleavage. Finally, they show that only 5' phosphorylated DNA is able to cause cleavage, and the other 4 lanes, where they tested single-strand RNA and non-phosphorylated DNA, cannot cleave. In Figure 3, the authors show that when transfected into HEC 293 T cells, they are able to get cleavage of a plasmid after a period of 8 hours of incubation. However, they suspected that Argonaute might have an endonuclease activity because they saw that when they were cleaving these and they sequenced them, that every time about 20 nucleotides were excised. So they tested whether they saw degradation of the plasmid after 72 hours by leaving the reaction on, and they didn't see that the DNA was degraded. Also, they show sequencing data that shows that even after 72 hours, there's no appreciable difference, so they conclude that the insertion and deletion pattern is actually a product of the cleavage itself and is not an endonuclease activity. They show in 293 T cells that using a GFP reporter plasmid and by Western blot readout, they were able to cleave the plasmid with 4 different pairs of primers, that is, guide DNA, showing that their construct works at least on plasmids in mammalian cells. They also show that the optimal length of nucleotide for the guide is 23 to 25. With all 3 of those, they got good cleavage. Figure 4, the million-dollar question. Can they cleave human genes? Well, what they show is that they can cleave the gene DYRK1, which has been used as a test for Cas9 enzymes in the past. DYRK1 is a dual specificity kinase. You're familiar with that. That means that it can phosphorylate serine, threonine, and tyrosine. Well, they show that with 4 different pairs of guides, they are able to get cleavage. And they do this by using the T7 endonuclease 1 assay. This assay is based on an enzyme from phage, T7, that can cleave only mismatched DNA. So what they do is they create double-strand breaks, they reanneal their DNA with normal DNA, and then the endonuclease will cleave, leaving these kind of products everywhere where the DNA double-strand break was effective. They show that this can be accomplished with 5 different genes, showing different patterns, but each time it effectuates a double-strand break. And they show with 4 different cell lines, they're able to get cleavage. They show that their guide DNA is very sensitive to any kind of mismatch. That is to say, it has to be exactly matched to the target sequence, which is good if you want a specific genome editing. So what they show here is all of these lanes are mutated in the guide DNA by only one nucleotide. And compared to the final lane, which shows complete cleavage, all of these have a 75% to 100% reduction in efficiency. Finally, they set out to prove equivalence or superiority over a Cas9 system. So what they show is for two genes here, they show that their construct is as good in terms of cleavage as Cas9 by the T7 endonuclease assay. And they show that for one gene, GATA4, which is very GC rich, their complex can actually cleave it, whereas the Cas9 system cannot. So here's Cas9, and here is NGAGO. Finally, with regards to genome editing, they test whether their Argonaute construct can induce double-strand breaks, which can then, via homology-directed recombination, induce novel DNA sequences into the genome. Using a GFP reporter construct and by flow cytometry, they can show that they got 12% efficiency at recombining by homology-directed recombination a new target sequence, thus showing that the Argonaute protein can be used for genome editing, in mammalian cells at least. To conclude, the RNA-guided endonuclease Cas9 has made genome editing a widely accessible technique. Similar to Cas9, endonucleases from the Argonaute protein family also use oligonucleotides as guides to degrade invasive genomes. Here we report that the Natronobacterium gregoryi Argonaute, NGAGO, is a DNA-guided endonuclease suitable for genome editing in human cells. NGAGO binds 5' phosphorylated single-strand guide DNA of 24 nucleotides, efficiently creates site-specific DNA double-strand breaks when loaded with the gDNA, and the NGAGO gDNA system does not require a PAM motif, as does Cas9. Preliminary characterization suggests a low tolerance to guide-target mismatch and high efficiency in editing GNC-rich genomic targets. Our second paper deals with designing a DNA polymerase with all mirror images of all its chiral elements. It's entitled A Synthetic Molecular System Capable of Mirror Image Genetic Replication and Transcription. So that would be DNA replication as well as DNA to RNA transcription. It's by Dr. Wang, and he's from the laboratory of Dr. Zhu Ting. They're from Tsinghua University in Beijing. It was published in Nature Chemistry, and it's somewhat chemical, but it's also a biotech paper. The first scheme details the experimental setup. So they have, basically, they're going to use natural amino acids are L amino acids and natural sugars are D sugars. So they're going to use D amino acids and L sugars in order to make a polymerase that's the exact mirror image. They're going to synthesize their polymerase chemically by making 3 peptide fragments on a synthesizer and then ligating them together. They chose as the target for their polymerase the smallest polymerase known in existence. It's from a virus, and it's less than 200 amino acids long, in comparison to the normal polymerase you think of, which is over 600 amino acids. So here they show the results of their purification. Here is a recombinant protein, that can be bought from the store. And here are their 2 attempts at making polymerases. One of them here is going to be the L polymerase, made from L amino acids, the normal kind, and the other one is their D polymerase. So as you can see, it's somewhat clean, but they've still got these smaller peptide fragments in their purified product. So I wouldn't call it pure completely, but it's at least the major component is the polymerase. And certainly it's probably the only one with any enzymatic activity. They show a circular dichroism spectrum, a CD spectrum, and as you can see, it's the exact opposite of the normal polymerase because it's the mirror image. And while it's not possible to say that it's the exact same spectrum, it at least shows that their product is folded properly into a secondary structure. It's not just a polypeptide. An assay of DNA replication. Figure 2 is all about their DNA replication. Here's the normal, wild type D sugar polymerase, D sugar DNA L polymerase, and this is going forward in the time direction. So you can see it extends a primer that's labeled with fluorophore into a full transcript. And this reaction was 4 hours, 37 degrees, so not too long. And then on this side, you've got your mirror image polymerase, which goes just as well, and with the same amount of stalling, I guess. Lower down, they show the amino acid, the nucleotide specificity of each, the normal and the mirror image polymerase. And as you can see on here, they're pretty much perfect. I don't see any really low fidelity in this at all. These reactions were 30 minutes at 37 degrees. Finally, they're showing basically the chiral specificity of their new enzyme. So they tested it with all different combinations of cross-chiral reagents. So the mirror image polymerase with normal primer, the mirror image polymerase with normal DNTPs, etc., and only the final lane where they're all chirally matched is able to transcribe. Figure 3, they show that the mirror image polymerase can function even in a racemic environment. So they've got each of the individual reactions. So normal wild type reaction, mirror image reaction, again labeled primers. And then this is actually not just a superposition of these two, but they have run a reaction with both of those sets of reagents in it. And you can see that it doesn't impair the efficiency of either polymerase. So there's no sort of cross-chiral blocking or competition by these mirror image reagents. Figure 4, they attempt to show that, well, it's not just replication that their polymerase can do, but the DNA that comes out is actually in a suitable form, an active form. So the way they do this is they find a piece of DNA that actually has catalytic activity in itself, a DNA-zyme. So the schematic of the DNA-zyme is that it folds into a certain secondary structure, and what it can do is it has some enzymatic activity, as you can see, and what it does is it cleaves a target in a zinc-dependent manner. So here, again, we've just got the classic wild type and mirror image. They're both able to elongate the DNA coding for this DNA-zyme. And they both have identical enzymatic activity, so this is without any reagents, with zinc and with magnesium. So both mirror image and natural enzyme can produce DNA, a DNA-zyme that's functional. Finally, they test the RNA transcription ability of their new mirror image product. In fact, they weren't certain that this small viral polymerase could be a transcriptase in the first place, but it turns out now these reactions look similar to what we've seen before, but this is RNA now, and it takes way longer. It takes 60 hours at 37. And what you can see is, especially on the mirror image system, there's quite a bit of stalling. So they see that the target has been elongated, but only by one nucleotide, they say. Yet, in the end, they can still get transcription from this enzyme. And just to prove it really is transcription, they've got their labeled primer product, and what they do is they digest that then with RNase A. So if it were DNA or something else, it wouldn't be digested, and you would see the band stay. Finally, they again test the fidelity of each nucleotide in these two, the natural and mirror image system. That's down at the bottom. And in this case, I must say, in both cases, it's a lot less faithful, because you can see in the RA reaction you incorporate some G, in the RC reaction you also incorporate some G, and in the mirror image it's even worse. In this case, in the U and A reactions you get quite a bit of C and G, but given that this is experimental and they weren't even sure that these polymerases had transcriptase activity, I'd say that's pretty accurate. In brief, the overwhelmingly homochiral nature of life has left a puzzle as to whether mirror image biological systems based on a chirally inverted version of molecular machinery could also have existed. I mean, chemically, there's no reason why it shouldn't. They're exactly the same. Here we report that two key steps in the central dogma of molecular biology, template-directed polymerization of DNA and transcription into RNA, can be catalyzed by a chemically synthesized D-amino acid polymerase on an L-DNA template. We also show that two chirally mirrored versions of the 174 residue African swine fever virus polymerase X could operate in a racemic mixture without significant enantiomeric cross-inhibition due to the activity of each other. Furthermore, we demonstrate that a functionally active L-DNazyme could enzymatically be produced using the D-amino acid polymerase. The establishment of such molecular systems with opposite-handedness highlights the potential to exploit enzymatically produced mirror image biomolecules as research and therapeutic tools. While I think this paper is very interesting and certainly well done and concise, I'm not entirely sure what the translational potential of left-handed DNA and right-handed amino acid transcription could possibly be. In their conclusion, they mentioned that it could be used to make bacterial factories, right? Biotechnology factories that would be resistant to any kind of virus, since they're unnatural. So that could be one use of this technology. That's all there is for this episode. Thanks for watching, and if I missed anything, please leave it in the comments.